So today I will discuss about complement system. So complement system complex, the C complex has total of 9 fragments, C1 to C9. So the fraction C1 is a calcium ion dependent complex which on chelation with EDTA forms three proteins, three protein subunits, which is C1Q, R and S. So in total it becomes 11 different proteins. So the fractions C1 to C9 is in the sequence of the cascading reaction except C4 because it comes after C1 and it comes before C2. It is like that. C4 is the exception. So the complement system, which is normally present in an inactive form in our body, but is induced by antigen antibody combination. So after it gets activated by antigen antibody combination, there is a formation, there is a cascade of series reaction is formed, and the preceding components act as an enzymes on the succeeding components, cleaving them into dissimilar fragments. The larger fragment ions join the cascade. The smaller fragments which are released often possess biological effects that contribute to the mechanisms like lysis of the bacteria and then opsonization of the bacteria, opsonization of the virus, neutralization and phagocytosis. It also removes immune complexes, and it amplifies the inflammatory processes. It amplifies the immunity processes. Basically, there are three ways by which complement can be activated. First is classical pathway, next is alternative pathway, and the third one is lectin pathway. The classical pathway requires a specific immune reaction for activation, but alternative and lectin pathway is independent of any antibody and antigen complex. These pathways are activated by reaction of complement proteins with surface molecules on microorganisms. C1, the deficiency of C1 is associated with a condition called hereditary angioneurotic edema. So it causes edema of the subcutaneous tissue of respiratory tract and alimentary tract, and it becomes fatal. The episode becomes fatal when it affects larynx and trachea. This edema is triggered when there is exhaustion of C1 inhibitor, and the cascade mechanism is started by formation of C1, C4 and C2, and the main mediator of this edema is the release of C2 kinase. This condition can be treated by the infusion of fresh plasma inside in the body, which helps in because this fresh plasma is a source of small amounts of inhibitors. So this fresh plasma is helpful, and also the patient can be administered with epsilon aminocaproic acid, because it is believed that epsilon aminocaproic acid or its analogues inhibit the activation of plasma enzymes.